Hello everyone. For this video, we're going to be talking about two very important enzymes involved in glycolysis, both of which are not reversible. So the first one we're going to be talking about is phosphofructokinase. As we can see within this picture I've displayed here, which I've actually taken from the first aid textbook, it's going to be shunting towards glycolysis. Now, how exactly is this enzyme stimulated? Well, it's activated using AMP, which, as we know, AMP means that there is a low amount of energy in the cell, and it's also stimulated by fructose 2,6-bisphosphate. Now, this concept is a little bit confusing, so we're going to come back to that. Now, it's also important to know how this enzyme is inhibited. If we remember that the purpose of glycolysis is used in order to make ATP and break down glucose, we know that if there is already enough ATP, then there is no need for this pathway to occur. So therefore, it is going to be inhibited by ATP. Also, citrate is a product of the TCA cycle, which is also involved in the production of ATP. So if that ATP is present, then we do not need the glycolysis to occur when citrate is present from the TCA cycle. So now let's get back to what fructose 2,6-bisphosphate and how exactly it's going to be enhancing this glycolysis pathway. So as we can see, fructose 2,6-bisphosphate is produced by an enzyme called phosphofructokinase 2. So what is this enzyme? Well, actually, it's not one single enzyme. It's called a bifunctional enzyme because actually PFK2 and fructose bisphosphatase 2 is actually one enzyme just with different activities. So these activities are going to change depending whether they're phosphorylated or dephosphorylated. And whether they're phosphorylated or dephosphorylated is determined by the hormones that are present. So let's talk about how this is going to occur. So as we see here, we have fructose 6-phosphate, which is an intermediate from glycolysis. And what's going to happen is that insulin, when there's an increased levels of insulin, that's going to cause a decrease in the activity of protein kinase A. And therefore, this bifunctional enzyme is going to be dephosphorylated. As a result, there's going to be an increase in fructose 2,6-bisphosphate. Now, if we follow the path and we see that fructose 2,6-bisphosphate stimulates PFK1, as we've also seen right here, then we see that this pathway towards glycolysis is going to be favored when there's going to be high levels of this molecule. Due to that, of course, the increase in insulin. Now, this makes sense, because think about it. Insulin is going to be released when there's high levels of glucose in the body. Therefore, since there's high levels of glucose, we want to break down this glucose. So that's why we are going to be going towards glycolysis rather than trying to produce glucose using gluconeogenesis. And it should also be noted that this molecule also plays a role in inhibiting fructose bisphosphatase 1 as well. Therefore, fructose bisphosphatase 1 as well. Therefore, further reducing the probability of going towards gluconeogenesis. Now let's talk about the role of glucagon. So if we keep talking about this bifunctional enzyme, and we know that glucagon's role is within the body, so let's think about what's going to happen. If you have a decrease in fructose 2,6-bisphosphate, then you're going to have more fructose 6-phosphate, and as a result, there's going to be less stimulation of PFK2, less inhibition of FBPase 1, and going more towards gluconeogenesis. So what's going to happen is there's going to be an increase in glucagon, an increase in phosphorylation that occurs due to the increase in protein kinase A activity, and therefore there's going to be less production of this molecule right here, therefore causing the pathway to be shunted towards the production of glucose. Now this makes sense because think about the role of glucagon. Glucagon's role is to increase the amount of glucose because there's a decreased amount of glucose within the body. So therefore, it's going to be shunting towards gluconeogenesis. Now this all makes sense, and I think this is a very important concept to understand because it seems difficult because of the varying roles of the different molecules involved. But the key enzyme you want to make note of is the key molecule is fructose 2,6-bisphosphate, and this bifunctional enzyme and how the hormones play a role in whether it's going to be phosphorylated and dephosphorylated, and that'll help you out big time. Now the last enzyme is going to be the last irreversible enzyme and also the last enzyme involved in the glycolysis pathway, and that's going to be pyruvate kinase. Now, pyruvate kinase is interesting because it's activated by actually a molecule that is more upstream within the glycolysis pathway, which is in a previous video that I just did. And that molecule that is going to enhance the activity of pyruvate kinase is actually fructose 1,6-bisphosphate. So this is what we call a feed-forward activator. Now the next thing we have to know about pyruvate kinase is going to be a similar concept to what we talked about here, is that it's going to be inhibited by ATP, because remember, this is going to represent high energy within the cell. Therefore, there's no need to be producing more ATP. Now, the next thing that's a bit tricky is why exactly does alanine inhibit the production of pyruvate kinase? Now, skeletal muscle, when skeletal muscle is deficient of glucose, what it's going to do is it's going to start to break down protein, and in order to create branched chain amino acids. Now, one of the byproducts of this degradation of protein is actually going to be alanine. So what this tells us is that the muscle is not receiving enough glucose. Furthermore, we know that alanine is actually going to travel from the muscle where it is broken down into the blood, back to the liver. This alanine then gets converted into pyruvate, which then undergoes gluconeogenesis to form glucose. So therefore, this alanine represents low levels of glucose within the cell because, as we can see, it's being used to create glucose, and the only reason it's being produced is because there isn't enough glucose being transported to the muscles. Therefore, it makes sense that if there's alanine present, we want to avoid breaking down this glucose into the glycolysis pathway. So that pretty much sums up these two videos that I made regarding the high yield enzymes in glycolysis. I hope you found them helpful, and good luck studying.